Hello, everyone, and welcome to today's eChalk Talk, hosted by Mercodia. You may submit your questions at any time by entering them into the questions bar of the GoToWebinar broadcast system. It is now my pleasure to introduce our speaker, Tanya Yarhirde.
Oh, thank you. So welcome to this webinar with the title Risk Mitigation in Bioanalysis to Maintain the Integrity of Drug Development Studies. My name is Tanya Yarhirde. I work as a senior scientist at Mercodia with assay development and validation of PK, biomarker and immunogenicity assays. I have previously worked at Biopharmaceuticals with binding kinetics and reagent characterization. I will in this session present some important aspects of assay development so that you will know what you measure, that you really measure the intended analyte. Ligand binding assays such as ELISAs are widely used in drug development and key decisions are taken from the data. It is therefore important that the results are reliable. So we will go through in this webinar how to choose binders that are specific and have a stable interaction with the analyte. We'll also talk about why it's important to have knowledge about the analyte and how to detect non-specific binding and minimize it. This is often a neglected subject. When you develop an ELISA, there is very many different things that you could optimize, and I will not go through all of them during this presentation. I will focus on the one in orange here. If you have experience from previous development, you know which steps that are most crucial to optimize, and you also have knowledge of good starting values that are most likely will work. We at Mercodia have more than 30 years experience of developing high quality ELISA kits, and we use that knowledge to provide bioanalytical services. And I will now share some of it with you. We start with how to choose binders. Here it was a project where we tried to develop an ELISA to measure AAPS, amylin. We have produced many monoclonal antibodies, and here we want to evaluate which ones we should use in building an ELISA. We did this study with surface plasmon resonance and Bioanalysis. Let's see here. You can see that we have a plot with the on rate and the off rates. And here you see the sensogram. The antibody is immobilized on the surface, and then we inject different concentrations of the analyte, AAPS, and follow the binding. And after we have injected the last concentration, there is a long injection with buffer to follow the dissociations. And we want to have binders that have a stable interaction with a slow off rate, and this is an example of a monoclonal that are stable. The antibodies in this region of the plot dissociate much faster, and we want to choose binders that have a stable interaction because then we minimize the risk of having an assay that could be sensitive to differences in the washing procedure. But we do not only want to have a stable interaction, it must also be a specific interaction. And here's another example. In this project, we wanted to measure glucagon, but there are other peptides that have very similar sequences. So we do not want to measure proglucagon, glycentine, and oxyntomodulin in this assay, only glucagon. And here we evaluated the binding also with the surface plasmon resonance. And we looked at antibody that binds in the N-terminus. And you can see that it has a stable binding between this antibody and glucagon. also similar affinity for binding to proglucagon. But glycentine and oxyntomodulin, they bind to the antibodies, but they dissociate fast, so they will be washed away in the washing step or in the ELISA. So they will not be measured in this with this antibody. But to have specificity so that we don't measure proglucagon, we also need an antibody that binds in the N-terminus. And so we do similar analysis with other antibodies and find antibodies that could bind to glucagon and not to proglucagon. And by combining these two antibodies into a sandwich ELISA, then we have a specific assay to measure glucagon and not the other peptides. So that was how we can early in the development how to choose binders. And now I will come into how knowledge about the analyte. This might seem obvious, but I have seen several examples where lack of knowledge have caused issues, and I will show you one example of that. So in this case, the sponsor came to us and needed a PK assay for measurement of a drug candidate in preclinical studies. There had been different challenges. A previous CRO had not, it has been very difficult to develop an ELISA. They prepared, calibrators in monkey plasma, just as it recommended in the guidelines that you should use the intended matrix, but also the blank matrix without any added drug had a high absorbance, even if the plasma was diluted 200 times. And when you spike in the drug, you see that there is very low sensitivity, not possible to measure the lower concentration here. So the sponsor needed to have this problem solved to be able to continue with the preclinical studies. So they came to us and we reviewed the previous data and tried to understand why it was such an unusual high response in the blank plasma. Was it some kind of non-specific binding or what was it? And to be able to solve this, we need to have knowledge about the analyte. And as a CRO, we do not have knowledge about all analytes, but the sponsor usually have a good knowledge about the analyte. So then there is a key to have a good communication with the sponsor, and also to have time to do some background research. And what we found about this analyte was that the drug candidate, the analyte, it's a recombinant human protein, and this protein is an endogenous protein that in normal plasma it's approximately less than 10 nanogram per milliliter. But it can be released from red blood cells and platelets during cell damage and inflammation in the body. It's also released from red blood cells and platelets if you take the sample and you get hemolysis, if the red blood cells or platelets are damaged, or if they can be damaged if you freeze thaw many times and so. So the quality of the samples will influence as a result. And since the purpose of the assay was to measure the drug candidate, not the endogenous protein, so then we could give a recommendation to the sponsor and to the study protocol how the sample collection should be done, so that they only measure the drug, not the endogenous protein that released from red blood cells and platelets. So now we have control over the sample collection. Next step is to have an assay to do the preclinical study. And that was done by modification of a commercial kit. And a commercial kit, if it's going to be used in a drug development bioanalysis, then there are some modification that is needed to fulfill the recommendation from regulatory guidelines. So the standards and the QC, instead of the ones that were in the kit, we produced standard QCs with the drug candidate. We used buffer matrix in the standard, and that was since since it also is an endogenous substance, it's not possible to find a blank matrix. It's recommended in the guidelines that you should use the intended matrix, but they also say that if it's an endogenous substance, it's you can use a surrogate matrix if you can justify it. And we justified it with the parallelism experiments. And by using a buffer matrix, we also saved plasmas from the animals to limit the amount of plasma used from the animals to fulfill the 3R recommendations. QCs were prepared in the intended plasma. And then by doing modification of the assay, we could also limit the amount of plasma that was used for each sample. So instead of having 200 microliter, we could use only 16 microliter. And that made it more suitable for studies with small animals with limited amount of plasma. And you can see the calibrator curve that we prepared in buffer with our modified, we optimized assay and the calibrator curve that was prepared in monkey plasma that were 200 times diluted from the previous zero. And actually let's see here, the concentration that they measured, the absorbance here in their blank plasma, it corresponds to a concentration that is at least 10 times higher than expected. So it could be that these samples were hemolyzed or had been freeze-thawed many times so that the endogenous protein had been released. During the development, we also saw that there was lot-to-lot variation of the commercial kit. So then we decided to use a single lot in the whole study. An alternative could have been that we could also have done lot bridging between the different lots. But the result was that we had a stable method with good data control, and we validated it to fulfill the recommendation from the regulatory guidelines. And we used it throughout different preclinical studies. So as I mentioned, it was achieved by having good knowledge about the analyte. And for this analyte, it was important to have control of the sample collection procedure. And then the modification of the commercial kit was done in this project. So now we turn to the next subject, non-specific binding. So we have talked about the importance of the knowledge of the analyte, but it is of equal importance to have knowledge about how to develop ELISAs. And everyone understands that it's important to have low non-specific binding, but still this is often neglected. And we will look also on how you could detect if there is any non-specific binding in your assay. Here in a sandwich ELISA, you want to measure your analyte, and you have a capture antibody and a detect antibody. But in ligand binding assays, you can also get cross reaction with the human anti-mouse antibodies, human anti-animal antibodies, heterophilic antibodies, or rheumatoid factors that could cause false positive. And this is especially in samples from patients with rheumatoid arthritis or other autoimmune diseases. But it can also be seen in healthy individuals, in some healthy individuals for a certain percentage of them. So when you investigate if it's non-specific binding, it's important that you look at your patient populations. You can also buy commercial human anti-mouse samples or rheumatoid factor samples and investigate if they give false positive in your assay. Non-specific binding could also be detected if you look at dilutions, and it can be seen as a lack of parallelism, but that is not always the case, that it shows a lack of parallelism. And then my favorite is to use a blank assay. And in a blank assay, you exchange either the capture antibody or the detect antibody with an antibody that is not binding to your analyte, so an unrelated antibody. And if you do that, exchange one of the antibodies, then if there still is binding, then you can be suspicious that it might be non-specific binding. And this is an example. Here it was a sponsor that has developed an assay for a biomarker, and they wanted us to optimize it. When we looked at the samples on a plate coated with the antibody that is specific to the biomarker in blue, and compare the same samples on a plate coated with an unrelated IgG in orange, you can see that for the QC samples that is a buffer with the biomarker, there is very low binding to the plate with unrelated IgG, as it should be. But on the samples that were plasma samples that were supposed to contain high levels of the biomarker, you can see here in orange that they also bind very much to the plate coated with an unrelated IgG. So that could be an indication of that this is non-specific binding. And I can also mention that the unrelated IgG, I try to choose one that has the same class and subclass as the antibody on the plate coated with the specific antibody. How to solve this if you have a non-specific binding? You could try to increase the minimal required dilution, but that is not always possible, especially if you have a biomarker. If you increase the minimal required dilution, then it could be too low, you get not enough sensitivity. Another way is to use blockers in your assay buffer to block this non-specific binding, and this could be commercial blockers or it could be irrelevant antibodies. And if we investigate this for this assay, we have in blue here the sample diluent from the sponsor and different samples from different subjects. And we compare with in orange where the samples are diluted with the sample diluent with blocking. And you can see that all these samples that were supposed to have high levels of the biomarker, when we use a sample diluent with blockers, there is very, very low levels measured. So the non-specific binding is removed, and then there seems to be no specific binding, the very low levels of specific binding. And when you use a blocking, sample diluent with blocking, it's important that you check that you do not block your specific response. So here we have the QC samples, and they were not affected by using blocking in in the sample diluent. So that is something that you always must check. Next slide. The 3 key factors that we have gone through so far are for developing reliable assays. It's careful selection of the critical reagent for high specificity and robustness, and that could be done with surface plasmon resonance. And we have talked about the knowledge, that it's important to have knowledge about the analyte, so you know what you measure. And finally, we have talked about how to check that the binding is not non-specific. And the goal of doing all this is to have correct data that could be used in the drug development studies. I will also give you some small tips and tricks that we think are important, and that is here at Mercodia we use monoclonal antibodies if possible instead of polyclonal to have a low lot-to-lot variations. We prefer also if possible to have pre-coated plates so you do not need to coat the plate before each run. So this if you have pre-coated plate, that reduces the time for each run and also better precision between the run. Here is if possible, we also try to use ready-made calibrators. In the recommendation from ICH M10, calibrators should preferably be prepared using freshly spiked standards, but they also state that if freshly spiked calibrators are not used, frozen standards can be used if you can show that they are stable. And that by using ready-made calibrators, we increase the precision between the run, so you don't have to do dilutions of the calibrators from high concentration before each run. So we try to use ready-made calibrators. And then there is a simple thing, color-coded reagent solution, and it improves the ease of use of the assay for the technicians. So it's the solutions that are colored, and then it's much more easier to see that you have put the right, but you have put solutions into the wells, and that it's more easy to take also the right solutions. And then finally, it's important to have control over the analytical life cycle, analytical procedure life cycle, the management of it. Yes, let's see. So that you can, if the assay is going to be used for long periods of times, and you will still get the same results in each run. And I think that was all from me. You are welcome to contact us if you have any questions, or if you want consulting or help with sample analysis, method development or validation for biomarkers, PK assay, immunogenicity and oligoassays. And as I said, we have more than 30 years experience of developing high quality ELISA kits, and we use that knowledge to provide bioanalytical services. So then if you can also, as they said in the beginning here, write questions in the chat. So, and now, do we have any questions?
Thank you, Tanya, for a wonderful presentation. My name is Esrat, and I will be moderating some of the questions that has come in. So what you spoke about the importance of knowing the analyte. What kind of information is needed about the analyte?
As I talked about in this webinar, I showed the example of that it was important to understand the biology of the analyte and the distribution between plasma and red blood cells and so. in this case, but it's also important to have knowledge about the physicochemical properties, such as if the analyte is very hydrophobic or heavily charged. Maybe you need to use low binding vials during the sample preparations. If the analyte forms dimer or multimers, then there is a risk that they dissociate during dilutions or incubations. So if you dilute many steps or they're standing a long time, if they dissociate, then you can see an increase in the concentration since they are dissociating. You should know if there are any homologous counterparts, something else that could cross-react, and also if there is information about already known information about the stability in the relevant matrix. That was.
Thank you, Tanya. And we have another question. That is, what is your experience with oligonucleotides? What are the challenges there?
Yes, for oligonucleotides, you must think about if you want to measure in plasma or in tissue. In tissues, the concentration are usually rather high, but if you should measure in plasma, there are very low concentrations. So then you would need to do the measurement with a method that has a high sensitivity. And for example, that could be an hybridization ELISA. The hybridization ELISA, on the other hand, is not so very good to discriminate between the full-length oligo and the metabolites that are shorter. So for that, LCMS may be a more suitable method. But when it comes to measuring in plasma and having a lot of samples, then the hybridization ELISA may be a better alternative, I think.
Thank you. And then we have this other question. Are there any implications for blocking non-specific antibodies in your assay?
Sorry, could you take the questions again?
Yes, of course. Are there any implications for blocking non-specific antibodies in your assay?
Yes, yes, that was the goal with blocking. If you go back to this non-specific antibodies, they must be blocked. Maybe we could come back to that question, but I'm not sure I understand it completely. Esra, do you have any more questions?
For PK assays, do you typically develop free drug assay or total drug assays, or both?
I think that we have most experience with developing total assay, but I mean, we could also develop for measuring the free.
Yes, so we have a question regarding to the hemolysis selected issue. Do you have any way to solve the problem of red blood cell contamination?
What we recommended was during the sample analysis, or during the sample collection, to prepare platelet-free plasma that included a more longer and more with a high centrifugation that was longer and with more speed, more rotation per minute, and also to not have many freeze-thaw cycles of the samples. And we also checked before running the samples that they were not hemolyzed.
Thank you very much, Tanya. That would be all of the questions in the chat. If you have any further questions, don't hesitate to contact Tanya on the email address provided. Thank you very much.
Thank you so much, and thank you to Mercodia for partnering with AAPS today. We look forward to seeing you all at a future eChalk Talk. You may now disconnect the webinar and have a wonderful day. Thank you.
Thank you.